Zebrafish are highly regenerative animals, with adults able to regenerate entire fins and heal injuries to the brain, heart, retina, and other tissues. They have proven to be useful in both forward and reverse genetic approaches for a growing community of zebrafish researchers. Adult mammals, on the other hand, have a very limited ability to regenerate damaged tissues. Once all mammals, including humans, reach maturity, they cannot regrow amputated limbs or fully recover from significant heart, spinal cord, or brain injury. Damage to the central nervous system is especially catastrophic because injury can lead to lifelong loss of function. The actor and director Christopher Reeve experienced the effects of traumatic spinal injury. The actor broke his neck in a horseback riding accident, shattering the first and second vertebrae and severely damaging the spinal cord. During his rehabilitation, Reeve eventually recovered sensory function in most of his body, but he permanently lost the ability to walk and was unable to breathe on his own. Christopher Reeve's limited recovery from spinal injury shows how adult mammals are unable to regenerate nerve cells. However, some adult non-mammalian vertebrates, including the zebrafish, can fully recover from significant nerve injury. Unfortunately, the cellular and molecular mechanisms behind neuron regeneration in these animals is largely unknown. Zebrafish studies have helped identify some of the mechanisms behind tissue regeneration, including the heart, muscle, and nervous system. Many research endeavors have used the zebrafish as a subject to try to understand the processes behind neuron regeneration. A study by Crony et al. Is particularly exciting because it identifies specific stem cells that contribute to nerve regeneration. The zebrafish experiment is unique because it concerns the origin of newly generated neurons. Most other studies focus on the growing population of nerve cells, rather than the origin of regeneration. By identifying the processes behind the zebrafish's remarkable ability to heal from nerve injury, we might be able to apply these processes for medical purposes. The zebrafish could hold the key for the treatment of injuries and diseases affecting the central nervous system in humans. So let's get into the experiment methodology of this study. The zebrafish subjects were all adults, at 6 to 10 months old and 25 to 32 millimeters long. First, the researchers had to simulate traumatic brain injury. All fish were anesthetized, and a cannula was pushed through a nostril to injure the telencephalon, a region of the brain that includes the cerebrum. The nostril served as a guiding tunnel to ensure that the same area was lesioned in each fish. 45 fish were stab lesioned in this manner, and a control group of 5 were sham lesioned, which means that the fish underwent the procedure, but the brain was not injured. Both the control and treatment group were kept under standard conditions. The chosen site of injury in the brain was known to not produce its own new neurons, since it lacked stem cells. So the experiment would determine whether the lesion received newly generated neurons from stem cells in other parts of the brain. In addition, the right hemisphere of the brain was left uninjured and served as an internal control. Over 90% of the lesioned fish survived the procedure. To monitor the response to brain injury, fish were periodically sacrificed and the lesion site examined under a microscope. In the day following treatment, a blood clot formed, shown in pink. The tissue took a spongy appearance due to edema, indicated by the yellow arrows. Edema is swelling due to a buildup of fluid and usually follows injury. Cell death in and around the lesion site was also observed. This next image shows that the zebrafish made a full recovery. There were no signs of chronic inflammation, apart from the occasional small cyst. The tissue architecture of the injured site was also completely re-established. Even when the left hemisphere, which was injured, is compared to the right hemisphere, which served as a control, there is virtually no difference in appearance. This graph shows that there was an increase in the production of glial cells in the injured hemisphere. However, production was regulated, preventing gliosis. Gliosis occurs in mammals following injury and involves the overproduction of glial cells, potentially leading to further damage and scarring. The fact that no glial scar was observed in the injured hemisphere shows that the zebrafish are able to regulate the production of glial cells following injury. However, an increase in glial cell production, or proliferation, does not confirm that neurogenesis occurred. Neurogenesis involves the production of brand new neurons from stem cells, which migrate to the site of injury and specialize their functions in order to form connections with local neurons. This has been thought to be impossible in adult mammals. Mammals are capable of producing glial cells from existing cells, but are unable to produce new neurons. This leads to an excess of glial cells, causing scarring and inflammation. BRDU pulse chase experiments were used to determine whether the neurons in the lesion site came from stem cells. BRDU is short for bromodeoxyuridine, a compound which can attach to the DNA of cells undergoing replication. The BRDU label attaches to cells undergoing mitosis, and the label is passed down to daughter cells. As the picture shows, BRDU-labeled cells were produced in the ventricular zone, which is known to contain stem cells. The labeled neurons then migrated to the lesion site. This confirms that the zebrafish are able to produce newborn neurons from stem cells in response to nerve injury. As this graph shows, there was an increase in newborn neuron population at the lesion site. Bromodeoxyuridine is an analog to the DNA base thymine, which means that it can replace this base. To perform BRDU substitution, DNA must be denatured, allowing BRDU to attach to the single strands expressing the chemicals associated with mitosis. The fact that BRDU is incorporated as a DNA base allows the label to be passed down to daughter cells. BRDU can be detected through staining, indicating areas where cell replication had occurred. The new neurons also succeeded in reaching maturity. The neurons expressed parvalbumin, a protein that is only expressed by mature neurons. Areas of parvalbumin expression are shown in pink. In addition, the new neurons were able to form connections, or synapses, with surrounding neurons. Synaptic expression is shown in red in this image of the lesion site. Areas of BRDU, parvalbumin, and synaptic expression were colored in the preceding slides through immunofluorescence. This technique utilizes the unique antibodies that are expressed by specific molecules within a cell. A fluorescent dye called alexafluor was conjugated to different antibodies associated with BRDU, parvalbumin, and synapses. The different dye conjugates bound to their respective antibodies. Areas of fluorescence in samples indicate expression of the target antibodies. So, we know that the zebrafish responds to injury by producing new neurons from stem cells, and that these neurons migrate to the site of injury, where they mature and form connections. But which stem cells are they? The researchers knew that radioglial cells were a good candidate, since they are known to be stem cells in infant mammals. In zebrafish, one type of radioglial cells are known to express the gene HER4.1. So, these radioglial cells were marked with green fluorescent protein, or GFP. As this image shows, newly generated GFP-marked genes were found at the lesion site, far from the ventricular zone where radioglial cells reside. So, to wrap everything up, this study found that radioglial cells expressing the gene HER4.1 are involved in neuron regeneration. Following injury, these cells produce new neurons that migrate to the site of injury, where they differentiate into mature neurons and become part of the local nervous network. The new knowledge that this study gives has important medical implications. There are currently two approaches to treating nerve damage: transplanting tissue to the injured site, or stimulating existing cells to induce regeneration. Zebrafish have strong potential to guide methods for regenerating neurons. However, in order to fully understand the mechanics behind neurogenesis, there need to be advances in gene targeting, chemical screening, and visualization techniques. Using the zebrafish as a guide, we may eventually be able to develop medical procedures for full recovery from traumatic nerve damage. I think that a potential experiment to further contribute to neurogenesis research would involve stem cell analysis in a tissue culture. The study mentioned that infant mammalian radioglial cells express an equivalent protein to HER4 called HE5. This potential study would evaluate the ability of HE5 expressing glial cells to respond to injury. The setup would be similar to the zebrafish experiment, except it would utilize tissue extracted from an infant mammal, such as a mouse. One tissue sample containing the target radioglial cells would be injured, and a control sample left uninjured to determine the extent of recovery. The radioglial cells would be GFP marked, and the site of injury examined for fluorescence, which would confirm that the stem cells had produced new neurons which migrated.